All right, we're going to go through a short sequence of slides to talk about how things are transported across a cell membrane. So starting off, here we have, once again, let's figure out what we're looking at. Here's a phospholipid bilayer. So this is the cell membrane. Oops. All right, this is the inside of the cell, and this is the outside of the cell. We're just using blue and yellow to symbolize that. Okay, so the first type of ways that molecules can move across a cell membrane is simple diffusion. And diffusion we've already talked about a little bit. It's how molecules want to spread apart, go from an area where they're highly concentrated to where there's not very many of them. So if I was to look at this diagram, we can see that there are all kinds of purple circles, whatever those molecules might be, on the outside of the cell, and not many on the inside. So I can draw what we would call a concentration triangle, I like to call this. So I'm going to pick purple here. So a concentration triangle is a simple way to show on a diagram where there is more of something and where there is less of something. So I'm going to draw a triangle like this, all right, long skinny triangle, and that's going to show you, I'll color it in purple. All right, so that's going to show you that there are more of these purple circles on the outside and less on the inside. And so using the law of diffusion, we can say that these molecules want to move from where there's more of them, which is currently on the outside, to where there's less of them, which is currently on the inside. Now, certain kinds of molecules can just move directly through the cell membrane. There's not many that can do this, but there are some. So examples of molecules that can move across the cell membrane without help, you should already know this list. It's things that are very small and do not have a charge. So like oxygen, carbon dioxide, urea is one. And then larger molecules, water too, sorry, that's a really important one, water. And there are some larger molecules that can also move across because they're hydrophobic. They have no charge. So things like, I think we said, the steroid hormones like estrogen, like testosterone, those kinds of molecules are able to cross the cell membrane because this greasy hydrophobic lipid bilayer is not a barrier to them, because they're quite happy hanging out in a hydrophobic zone, because they themselves are also hydrophobic and non-polar. So the large cholesterol, estrogen, testosterone, nicotine, those kinds of molecules can cross the cell membrane. And this is an example of simple diffusion straight through the lipid bilayer. All right, so that's our first kind of transport. All right, so our next kind of transport, this is called facilitated diffusion. And in facilitated diffusion, we have a facilitator. A facilitator is a helper, right? So we are still doing diffusion, which means things are moving from where there's a lot of them to where there's not very many of them. So if I'm going to draw a concentration triangle for these orange triangles, it's going to look like this, right? We've got, oops, that's a bad triangle. Let me start over. We've got a lot of them on the outside and not very many of them on the inside, right? I'm artistically challenged right here with my triangles. There we go. Right. So there's more of them here and there's less of them here. So the direction that they want to move is from more to less, because they're a solute and they want to go from more to less. And these triangles are getting into the cell through a channel or a tunnel, which is this protein that we can see here. So this is our transport protein right here, this purple blob, and it's allowing these orange triangles to get through. So what could these orange triangles be? Well, there's a whole list. Remember we said any kind of ion, so Na+, K+, anything largish with a partial charge, like glucose, like starch, that's a really big molecule. Things that are like amino acids. These are all kinds of molecules that are too big and have charges or partial charges, or tiny but have charges, to pass through directly through the bilayer. So they need this helper, this channel. But if you provide the channel, the right channel, they can go through, and they're going to go based on their concentration gradient. They could go from outside to inside. They could go from inside to outside if the concentration gradient was flipped. So if we had more triangles here, lots and lots and lots and lots of triangles, and not very many up here, then they would go that way instead. The direction of transport would be controlled by the concentration. Where is there more, where is there less? This is facilitated diffusion. It's diffusion, but it's with a helper of the tunnel. All right, last but not least, here's another example. Once again, here's our phospholipid bilayer. Ooh, trying to change color, hold on. Here's our phospholipid bilayer. And here's the outside, and here's the inside. And we are moving these green squares into the cell. But what's different here is that there are already a lot of green squares inside the cell. So if we were to draw a concentration triangle for this diagram, it would look like this. So I'm going to draw a big skinny triangle going this way. Right? So we're showing way more green triangles, green squares on the inside and way less on the outside. So if you were to leave these green squares up to their own devices, they really want to go this way. This is the way they want to go. But your body doesn't want that. Your body wants to do the opposite. Your body wants to accumulate big green squares on the inside. So to make more green squares come to the inside, we're going to have to force them in. And that's going to take effort and energy, and that's what this ATP is showing here. And so this is actually a pump. And this is the protein acting like a pump, pushing green squares into the cell opposite to the direction that they actually want to go, so that we can accumulate a big old puddle of green squares, whatever that might be, down here in the cell. And this is called active transport, and it's active because you're using energy or effort to get it done. It's not going to happen on its own. So to summarize all these slides, here you can see them all at once. So we have the diffusion without a helper and the diffusion with a helper. And in both these cases, if I was to draw a concentration triangle for the solutes, for the purple circles or the orange triangles, the triangle would look like this. And these are examples of passive transport. This is just going to happen in this direction on its own without any help or any pushing. It's just the way the molecules will go by nature. The last example with the green squares is active transport. It takes action or energy to get this to happen. You've got to push it or force it, like pushing something up a hill. And so if I draw the triangle on this side, it's going to be flipped around like that, showing more green squares down here and less up here. So once again, this is like rolling something down a hill. This one, it'll go on its own. This one is like pushing something up a hill. It won't go on its own. You've got to push it. That's why it's called active. So active transport and passive transport, and some examples of how they function in cells.